everyone. Thank you for joining us today for this presentation. In today's presentation, we will be covering an overview of antimicrobials and their effects on product testing, understanding antimicrobials and how they can impact your product testing. My name is Deveny. I am a scientific analyst for STERIS working at our Brooklyn Park, Minnesota lab in the US. Supporting a lot of our microservices laboratory testing. Today, we will be covering a variety of topics, and I'll be walking us through some fundamental principles of antimicrobials and some common antimicrobial agents.
And hello, my name's Craig Miller. I am from the Thorne Laboratory in the UK for STERIS. I assist in the laboratory testing in Thorne. And I will be covering the importance of medical device industry and the testing for antimicrobials, a short overview of laboratory method suitability, and myself and Deveny will go through some case studies for some examples of antimicrobials and their testing, and then a short summary at the end to overview what we've discussed.
To get things started, we need to talk about what is an antimicrobial. The term antimicrobial refers to the effect on microbes that is observed. Antimicrobials destroy microorganisms or prevent their ability to replicate and grow in the presence of that antimicrobial. This is a very broad category that encompasses many different types of antimicrobial agents, including but not limited to molecules, polymers, the byproduct of another microorganism, and elemental materials such as silver or copper, etc. Some antimicrobial components are non-selective, which means that they have an impact on all microorganisms that are exposed to that antimicrobial. Others may be selective, targeting certain microorganism types while having minimal or no impact on others. And this is primarily due to the mechanism of that interaction between the antimicrobial and the microorganism. So that selective category may include just targeting certain types of microorganisms such as fungi, bacteria, or viruses. So we'll start by walking through some definitions as well, just getting some of that out of the way. So bacteriostatic or inhibition, so this is the prevention of bacterial reproduction and growth that does not kill the microorganism outright, just inhibits their reproduction. Method suitability. This is an assessment of the antimicrobial activity of the product during testing. Inoculation. The process of introducing microorganisms to a system or a product. Leaching, the loss or extraction of certain materials into a liquid. CFU, this is colony forming units. Neutralization, stopping or reducing the level of inhibition demonstrated by the product. And another acronym, MSDS, Material Safety Data Sheet. So getting into the mechanism, just like larger multicellular organisms, bacterial, fungal, and viral microorganisms have normal functions that can be interfered with, interrupted, which will lead to cellular damage and destruction. Antimicrobial components interfere with one or multiple of these functions. So that might include interfering with the cell wall, the ability of that organism to replicate DNA, metabolism, or protein synthesis. So cell wall, that's an important barrier in a microorganism. The internal environment of that organism must be balanced. A broken cell wall barrier disrupts that balance, which leads to microorganism death. DNA replication. Replication of the genetic code for the microorganism is a critical growth step. Interrupting that impedes the ability of that organism to replicate. Metabolism. The ability to take nutrients and convert them into energy for growth and reproduction, again, an essential step. And protein synthesis. Proteins are essential for normal function, and they support several of these processes we've already discussed. Growth, metabolism, and DNA replication are all supported by the creation of additional proteins in the microorganism. Interference with protein synthesis may have a ripple effect into many of these other processes. Different groups of microorganisms have evolved to function in slightly different ways, and these differences dictate which species are susceptible to the inhibitory or destructive effects of a particular antimicrobial. So we have some categories of different antimicrobials. As we discussed earlier, that is a broad term that encompasses a wide variety of product types. Some examples may include a disinfectant, which is often applied to a surface or included in a liquid solution. Antiseptics, which is applied to tissue to reduce the likelihood of infection from opportunistic pathogens. For an example of this is a hand sanitizer. Antibiotics, administered orally or by injection to treat or prevent infection. That also might include a material component of a medical device, molecular compounds found in aqueous products. We've got some examples of disinfectants and antiseptics on the screen, such as asbesticide, STERIS products, Spore Cleanse, or Septahol. So, why do we care about antimicrobials in medical device when many products undergo terminal sterilization processing before distribution anyways? Inclusion of antimicrobials may reduce product bioburden. Bioburden testing is key for ongoing monitoring of manufacturing and routine sterilization, in addition to establishing a validated sterilization process. Bioburden testing monitors the presence of contaminants on product pre-sterilization, and the inclusion of antimicrobial components in a medical device may help reduce the number of microorganisms present on a product before the sterilization. Having this extra level of control of your bioburden before sterilization helps to prevent fluctuations in the amount of product bioburden present from batch to batch. So this might reduce the radiation dose or the EO concentration needed to achieve the appropriate level of sterilization. Supply chain issues are also an ongoing challenge in medical device manufacture. Changes within a supply chain may also lead to unanticipated change in the number of contaminants present in the product, and the inclusion of antimicrobials may help prevent severe fluctuations from these supply chain issues that are often outside of the control of the device manufacturer. Again, reducing the bioburden present on a product reduces the radiation dose or EO concentration needed for effective sterilization. This in turn may lead to lower processing costs and shorter processing times. For example, less time required for EO off-gassing after that dose has been applied. There are even benefits after the product has been sterilized and shipped. Once removed from the sterile packaging, the product may again be exposed to microbial contaminants during the end use of the device. The inclusion of antimicrobials that reduce the amount of contamination during product use may help prevent that from causing a downstream infection in a patient receiving treatment. For example, biofilms that may develop on an implanted device are a high concern for patient safety, and antimicrobial elements may reduce the likelihood of biofilm formation. Even when not intentionally introduced, antimicrobial elements may be present in your product, as many materials used for medical device manufacture are inherently antimicrobial, for example, copper or silver. So now we're going to walk through some common antimicrobial agents, just a few examples of ones you might see, their mechanism of microbial inhibition or destruction, and some methods that the laboratory might use to neutralize, just to get an idea of the widespread range of antimicrobials that we might encounter on the laboratory end. So alcohols, one we're familiar with, commonly found in antiseptics or disinfectants. Some examples include like an isopropyl alcohol, ethyl alcohol, or propanol. The mechanism for these is often membrane permeability. So again, getting through that protective coating on the microorganism, permeating in and impeding that internal balance. And a common neutralization method for getting rid of this effect is, and allowing the recovery of microorganisms, is dilution, diluting that effect. Silver is another common antimicrobial agent, which may often be included in a coating product in a device, even in ion form. So even if it doesn't look silver to the eye, there may be those ions, so a form of that silver included in that product. There's a lot of mechanisms for this antimicrobial. It may impact the cell wall, cellular respiration, metabolic function, and DNA replication. And neutralization is thioglycolic acid. Aldehydes, this is a very common preservative. Some examples of that include glutaraldehyde, formaldehyde, and OPA. Again, many mechanisms. This is common for a lot of antimicrobials. You'll see multiple different impacts due to the connections between a lot of those cellular functions. So cell wall disruption or lyse, busting open that cell wall, damage to the contents of the cell, the bacterial, viral, or fungal organism, and impacting some of their critical protein and amino groups within that organism. So some neutralizations include thiosulfate, bisulfate, dilution, or glycine. So again, a couple of options for this one. Quaternary ammonium compounds. So quaternary ammonium compounds are a category of chemical that contains an array of different chemical compounds. It's a very broad group, commonly used as preservatives or a component of disinfectants, including ones you may have used at home. In medical device, they are frequently included as a part of antimicrobial coating products. Mechanism is generally just, again, getting through that membrane, impeding the ability of the organism to have balance between the outside environment and the inside environment, damaging the contents, the cellular contents of that organism, and some neutralization methods may include polysorbate or lecithin. Parabens. These are typically used as preservatives in cosmetic products. We've got a long list. There's many different parabens in this category. Again, mechanism may include membrane disruption, DNA, RNA inhibition, and polysorbate is typically a good neutralizer. And again, the common antimicrobial agents just offer an example of just a wide array of components that might be included in a product, and how diverse they might be. Now we're going to move on to laboratory testing, getting into some more details on how this might impact the laboratory testing that you might see for your product. So passing this over to Craig to walk us through some of the standards and, again, working into some of that laboratory testing.
Thank you very much, Deveny. So we're going to start off as we move into more associated medical devices by covering some of the standards in Pharmacopeia that are referenced throughout. We're going to firstly start with ISO 11737 Part 1. This is the ISO for bioburden testing. And in conjunction with this, we have ISO 11737 Part 2, which is about the sterility testing of products. In conjunction with both of these, we will refer to the ISO 11137 Part 2, and this is for the establishing of sterilization doses for healthcare products. As well as the ISO standards, we will refer to some Pharmacopeia. We have two of the main ones we'll reference will be the United States Pharmacopeia and the European Pharmacopeia, USP and EP respectively. So for sterility testing, we'll be referring to USP Chapter 71 and EP Chapter 2.6.1. And for bioburden, we'll be referring to USP Chapter 61 and EP Chapter 2.6.12 for the bioburden testing. Now that we've got through the standards, in our previous Why Do We Care slide, we discussed how the inclusion of antimicrobials are of a benefit to products that will undergo terminal sterilization. However, despite these benefits, there are some concerns in regards to the testing processes used to validate said terminal sterilization processes. This is due to the antimicrobial agents possibly creating a false negative result, which may then negatively affect the sterilization validation, which we'll cover in a few slides' time. As per the ISO standards 11737 Part 1, which is our bioburden, and Part 2 for our sterility, as well as the applicable Pharmacopeia chapters, these detail that a method suitability shall be demonstrated. And what we mean by method suitability is Bioburden recovery efficiency. This is detailed in our ISO 11737 Part 1. Our bioburden method suitability, which is touched upon in ISO 11737 Part 1, but is covered in a bit more detail within the applicable Pharmacopeia chapters we've just discussed, as well as bacteriostasis and fungistasis, which is touched on in ISO 11737 Part 2, but again is covered in detail within the applicable Pharmacopeia chapters. The purpose of the method suitabilities are to determine if the product is being successfully neutralized for those that have known antimicrobial substances and therefore their antimicrobial action, or that the method for testing being applied is suitable and does not lead to a leaching of an antimicrobial substance into the testing process. With the addition of antimicrobials, the demonstration of this method suitability can be more challenging, and it is essential to determine that the method is suitable. So now we're going to have a short look at each one of these method suitabilities and demonstrate the effects of the antimicrobials, and then we will come back to their importance in medical devices. The first we'll cover is that of the bioburden recovery efficiency. This is the determination of the ability to recover microorganisms from the product that's being tested. So how many microorganisms that can be removed during the testing process. This is completed via two methods. We have repetitive recovery and product inoculation. Repetitive is the use of the natural bioburden, the bioburden that is on the product that is being placed there during its manufacture, whereas the inoculated is the addition of known organisms to the product, usually on products that are low in natural bioburden. We will focus on inoculated on this short overview. Ideally, the organisms are inoculated onto their product to replicate the natural bioburden so that they're fixed to the product to replicate that washing process as accurately as possible. But this can also be inoculated into the extraction fluid for absorbent materials such as dressings. This inoculation, in the presence of a direct antimicrobial, may lead to a reduction of the viable spores and create a false recovery efficiency. And what I mean by that is one that is lower than is giving you lower counts, not based on how well it's been washed off, but due to the antimicrobial ability. Therefore, we want the neutralization step to occur either before the inoculation or as short a time as possible after inoculation to reduce this effect. The product will then go through the same extraction process as the proposed method for the product, and the results are gathered. You're looking at your plate counts against your control counts. There is no defined acceptance criteria for this testing. It is a very variable, depends heavily on the type of product, and this variance is detailed in ISO 11737 Part 1, Section C.5.1. Now we will discuss the method suitability, which we are going to refer to as inhibitory substance screening for this webinar. And this is the assessment to demonstrate the lack of inhibition of growth of microorganisms. The purpose of this testing is to ensure that the extraction process that is undergoing for the product and that testing methodology is sufficient to neutralize any antimicrobials that may be present within the product, or even compounds that may have leached out during the extraction process of the testing. We can assess this possible leaching or antimicrobial effect by adding the neutralizing agent or a dilution before the inoculation. This ensures that if there are any antimicrobial substances or compounds present, they're able to be neutralized before the organisms are introduced and inoculated. The organisms used can be as per a laboratory specific options or even customer defined based on requirements that could range to expected organisms within your system, such as gram-positive cocci, gram-negative rods, or even further regulatory requirements as applicable. Again, it follows the same extraction process as the proposed method. The results are gathered, your plate counts against your controls, and within this ISO standard, there is no defined acceptance criteria as to what is acceptable levels of inhibition. However, in our next slide, we will discuss the Pharmacopeia testing for this inhibition, which does have some. Both the United States and European Pharmacopeia detail the process for this bioburden method suitability in USP Chapter 61, as well as EP Chapter 2.6.12. The Pharmacopeias give details on how to demonstrate a suitable method depending on the physical characteristics of the product. So they have a list. If your product is able to be tested as described, you can use those tests, but also gives guidance if you're looking to develop a suitable alternative. As with before, it is used to determine the ability of the method to neutralize any potential antimicrobials within the testing process, but provides greater specifics than that of the Bioburden ISO standard. The Pharmacopeias provide a list of organisms that are to be used for the assessment of the method suitability. They have a set list, and with that set list of the organisms, they have incubation conditions to be utilized as well for each of the respective organisms. That includes agars to be used and their incubation temperatures and times. Whereas the ISO standard does not give an acceptance criteria to be used, the Pharmacopeias provide one of greater than 50% recovery when compared to that of the control counts. And the control counts are those that are inoculated into the same type of extraction fluid but are absent the product. That means if there was a greater than 50% reduction of the sample counts compared to that of the controls, the current method being used for the product would not be deemed suitable as per Pharmacopeia, and further actions would be required. Whether that be further dilution of the product to aid in the neutralization via dilution, or even an addition of a neutralization agent, if there is currently none being added, or increasing the concentration of already present neutralizing agent. With this short overview of bioburden method suitability, we'll now just cover the sterility method suitability. This is more commonly known as bacteriostasis and fungostasis, or BNF. And as indicated by its name, it is assessing the product's ability to either inhibit or stop the growth of bacteria and fungi. ISO 11737 Part 2 refers to the method suitability and the requirements that shall be performed to display the effectiveness of whatever neutralizing system is in place. The standard will then also refer to the relevant pharmacopeals for guidance on how to actually perform these tests, such as our USP 71 and our EP Chapter 2.6.1. The product would be added to a culture medium. This is often soybean casein digest medium, which is also referred to as TSB, but can also be fluid thioglycolate medium if there is cause to suspect anaerobic organisms and you want your method suitability to cover those organisms. The samples are inoculated with known amounts of the organisms that are required and then left to incubate for the appropriate time frame. As with when it comes to sterility testing, the growth can be assessed with a visual check on the containers, such as in the photo in the slide. You can see for most of them there's a clear turbidity, and with the two closer ones there is a slight hint of turbidity. But they can also be subcultured using an appropriate agar or method if the product obscures the turbidity that is hoping to be seen. So, with those brief overviews of the method suitabilities, for each of those testings that may be required for your product. There is the big question: why is it important for the testing to show growth? Surely, if there's no growth in the testing, it's good for our products and the patients further down the line. As a little note, we will be mainly referring to medical device testing in this slide, but the information can be applied to other product types. So firstly, we're going to look at the bioburden. Bioburden, as we've already discussed, is a key factor in the establishment for a routine dose in the 11137 ISO standard. We use the average bioburden to calculate the verification dose as part of this validation. If the method suitability isn't performed correctly and there is a reduction due to this die-off observed in the bioburden average for the product, this may actually lead to then a reduced verification dose being applied. And with this reduced verification dose being applied, it's then not applicable to the correct number of natural organisms that will be found on your bioburden for your product. And then this could lead to a failure of the substantiation of the routine dose as per the ISO acceptance criteria, as the organisms and the number and types of organisms would not have had an appropriate verification dose being applied. And then you will see this via positive sterility samples occurring. And when it comes to ethylene oxide, if we're looking at ISO 11135 Annex A, having the ability to demonstrate a more accurate bioburden count can be useful for the reduction of the ethylene oxide use and maintain that confidence in the sterilization process. If we now look to sterility for both irradiation and gases, there is a section A.7 in the ISO standard 11737 Part 2, which provides an overview of the risks of false negatives. The main takeaway of which is that false negatives can affect the interpretation of the data negatively in the validation. This has the unfortunate effect, if it's appropriate, of giving the sterilization process and the sterilizing agent as seeming more effective than they are, and leading to a possible risk to end users of the products. So as can be seen, usually in more relation to radiation standards, but is applicable throughout, there is a correlation between the ability to recover organisms within bioburden testing and sterility testing, and so the method suitability for both of these, where applicable, is very important for the validation of terminal sterilization. In the next few slides, we're going to have some examples of real-world antimicrobials and their testing involved and the work required to ensure valid and appropriate results for our customers. I'm going to pass over to Deveny, who's talk us through our first one.
Thanks, Greg. In our first case study, we'll be looking at a zinc antimicrobial. And so this particular product, and antimicrobial components are essential in non-sterile products. And this particular product was non-sterile and had a very strong antifungal component present in that product. So this is critical for multi-dose products, as it may reduce inadvertent contamination during use of that product. So during the method suitability for this test, we recovered bacterial organisms with minimal neutralization needed. However, due to the strong antifungal antimicrobial present, we had difficulty recovering the fungal organisms, and we required extreme dilutions to have that recovery present to demonstrate that we could recover those organisms. So dilution as neutralization, it's effective, but as the dilutions become greater and greater, it increases the limit of detection of the method. So as we dilute the antifungal that's present in the product, we have a harder time saying that saying how few are actually present when routine testing occurs. So in product development, this highlights the importance of finding the right amount, enough to inhibit growth without toxicity to those microorganisms. So effective antimicrobials are inherently toxic, and the concentration of these components should be minimized. Moving on to our next case study, passes back to Craig.
Thank you very much, Deveny. So in our second case study, this is of a device that was undergoing its routine dose establishment as per the ISO 11137 Part 2 standard for VDMAX 25. It was a new product, and from both the customer assessment and the laboratory assessment of the material safety data sheet, there was a clear worry that the pH of the testing was going to be an issue, as there was the presence of an acid within the product, and that may have an effect on the organisms during the method suitabilities. In advance of the testing, we received some samples from the customer for the project, and during the assessment of these samples, it was noted that the level of extraction fluid that was proposed for the product would be sufficient for the neutralization of the pH. It brought it down to neutral levels. So with that sort of confidence that we had handled the pH, we move then to the testing process of the method suitabilities. And with the BNF and the recovery efficiency, we got appropriate growth that was deemed successful by our customer. However, when the testing of the inhibitory substance screening was occurring, the results showed that there was no growth of certain organisms, which was quite surprising considering the relative success of the previous testing. The discussions were then had with the laboratory and the customer to try and come together to think of any other factors that may be within the product that they may know from manufacturer, but however, no obvious issue was found, and so it was decided to repeat, but with an increased dilution to remove the unknown antimicrobial action, as this is a generic method of neutralization and usually quite successful. Unfortunately, the results were the same, with the bioburden recovery efficiency results coming back within the desired specification for our customer, and again the inhibitory substance screening, it was found to have no growth with the same organisms. On this news, we went back and had further discussions with the customer, who did a more in-depth look at their product, and one of the results was that the solution within their product had trace amounts of silver, and it was thought to not be enough to have an effect, but this small amount of silver had enough of an antimicrobial effect on the growth of some of the organisms we were testing. With that knowledge, we went back to the product and the testing methodology. We added sodium thioglycolate to the extraction fluid, and from that point all the method validations were successful, and we were able to validate a minimum routine dose of 25 kg for our customer. So I think this case study likes to highlight that even small amounts of antimicrobials can have an effect when it comes to the testing of the products. And for our third case study, we have polyhexamethylene biguanide, or PHMB. This is a known antimicrobial polymer, and it's added to products for its known antimicrobial properties. It's there as a disinfectant and an antiseptic. Before the product was submitted to us for this routine dose establishment, discussions were held with the customer to allow for an agreed methodology to attempt to neutralize the PHMB, as it's known to be quite difficult to neutralize. We were able to complete multiple trials looking at different concentrations of different neutralizers and methodologies to determine what worked for the PHMB within the product. And the product of this trial work was we were using lecithin and polysorbate at high concentrations, as well as an addition of further dilutions after the extraction process, and that was allowing us to obtain appropriate results. And I think this example does show that as antimicrobials continue to improve and evolve, there may not be a simple solution to neutralizing an antimicrobial, but through the use of multiple methods of neutralization and sort of working together with our customer, it will allow for successful testing. So hopefully throughout this presentation and demonstrating our case studies, there is an importance about having open discussions within with our testing laboratories and our customers, and it does help to reduce the chances of any issues that may arise. So some examples of like communication is before samples are submitted for their project, providing a detailed information of your product composition, but also working with that leads on to having an open dialogue with our customers in STERIS, where we highly promote this, and if there is any possible antimicrobials that wish to be discussed, you can contact your local STERIS representatives, and all that discussion can help smooth things out at the start. But also in addition, the provision of extra samples for a potentially challenging product helps smooth out if there is any issues in the future, that the testing process is delayed as little as possible. So to finish off, we're going to go over a short summary of some of the advantages have advantages for both the customers and the laboratories. One of the main ones is reduces the likelihood of issues and repeats. So having this communication and knowledge in advance, it's more likely to pass first time, and therefore you're less likely to be hindered by repeats. This is allowed due to the laboratory being able to incorporate the best neutralization method from the start of the process, and so it is more tailored to the products. And a byproduct of this is that it should speed up testing. If there are less issues and repeats, the results are more likely to be achieved within their established turnaround times. An advantage that is always welcome is no unexpected costs. One of the main causes of unexpected costs is repeats. If something doesn't go well the first time, we then have to cost again for the second one. So by having this initial open dialogue at the start and this information exchange, we can help reduce that by being more specific with our neutralization. And for me, I think one of the most important ones is confidence in your results. By working together with the testing laboratory and in conjunction with our customers, we're able to ensure that the product is getting tested as appropriate as it should and neutralized as it should with these method suitabilities. And that gives you the knowledge that your test results are correct. That gives you empirical evidence for your terminal sterilization validation that what is being performed is correct and substantial for what you require. So I'd like to say thank you for myself, Craig Miller, and
thank you as well from Deveny. Our contact information is available on this slide. Feel free to reach out and contact us with any additional questions.